Welcome to G Fun Facts Online. Today, we're diving deep into the microscopic battleground of the brain, where a new dawn is breaking in the fight against Parkinson's disease. Parkinson's affects millions globally, gradually taking away motor control and cognitive function. For decades, we've known the primary culprit is a protein called alpha-synuclein, which misfolds and clumps together, poisoning crucial brain cells. Current treatments manage symptoms, but can't stop the disease's relentless march. But now, scientists are on the verge of a breakthrough, using custom-designed molecules called peptides to stop this toxic process in its tracks. Imagine a tiny, precision-guided missile that can seek out and neutralize the very source of Parkinson's. That's the promise of peptides. Let's explore how these microscopic heroes work. Parkinson's is the world's second most common neurodegenerative disease. Its visible symptoms, like tremors and rigidity, are the result of a devastating loss of dopamine-producing neurons in a brain region called the substantia nigra. The therapies we have today focus on replacing that lost dopamine. They can be life-changing for a time, but they don't halt the underlying destruction of brain cells, and long-term use can bring on its own set of debilitating side effects. This has created an urgent need for therapies that modify the disease itself, not just the symptoms. And the prime target for such a therapy is the misfolding of alpha-synuclein. In a healthy brain, alpha-synuclein is a flexible, helpful protein. It plays a key role in regulating the release of neurotransmitters, including the very dopamine that's so critical. It's a vital part of our neural machinery. But in Parkinson's, this protein turns treacherous. It starts to misfold, changing its shape in a way that makes it sticky. These sticky, misfolded proteins begin to clump together, forming what are known as oligomers. For a long time, scientists thought the large, visible clumps called Lewy bodies were the main problem. But now, evidence overwhelmingly points to these smaller, earlier-stage oligomers as the most toxic species. They are the real assassins, disrupting cell functions and triggering the death of neurons. As these oligomers continue to aggregate, they form larger, insoluble fibrils, which eventually become the Lewy bodies seen in post-mortem brains. It's crucial to understand that this protein aggregation isn't just a side effect of the disease. It is a key driver of its onset and its progression. The central challenge for researchers has been how to stop this aggregation without interfering with the normal, beneficial functions of alpha-synuclein. It's like trying to stop a specific type of car from crashing without closing the entire highway. This is where peptides are emerging as a uniquely powerful tool. Peptides are simply short chains of amino acids, which are the building blocks of proteins. You can think of them as small pieces of proteins. Their appeal in fighting neurodegenerative diseases comes from their incredible specificity and potency. Unlike many small molecule drugs that can have off-target effects, peptides can be designed with high precision to bind to a specific region of a target protein. This allows for a tailored, strategic attack on the alpha-synuclein aggregation process. Scientists are developing peptides that intervene in several brilliant ways. One of the most exciting strategies comes from researchers at the University of Bath in the UK. They have engineered a peptide that essentially acts as a molecular chaperone. In its active, healthy state, alpha-synuclein folds into a helical shape, which is critical for its job in dopamine transport. The peptide designed at Bath binds to alpha-synuclein and locks it into this healthy, helical form. It physically prevents the protein from misfolding and starting down the toxic aggregation pathway. This preventative approach is incredibly promising because it could potentially stop the disease before it ever takes hold, especially in individuals identified as high risk. When tested in a worm model of Parkinson's, this peptide not only reduced the buildup of toxic alpha-synuclein clumps, but also improved the worm's motor function. To make the peptide more durable inside a cell, the researchers cleverly stabilized its structure using chemical bonds called lactam bridges. Another ingenious strategy is to use peptides to cap the ends of the growing alpha-synuclein fibrils. Imagine the fibrils are like a Lego tower being built one brick at a time. These capping peptides are designed to form a specific shape called a beta hairpin that fits perfectly onto the end of the fibril. By binding there, it blocks any more misfolded alpha-synuclein bricks from being added. This approach effectively puts a lid on the aggregation process, halting the formation of larger, more damaging clumps. A third, seemingly counterintuitive strategy involves redirecting the aggregation process altogether. The idea here is that not all aggregates are equally harmful. Some peptides are being designed to force alpha-synuclein to clump into amorphous, less structured aggregates rather than the highly organized, toxic fibrils. By diverting the protein down a less harmful pathway, it might be possible to sequester the misfolded alpha-synuclein into a form that neurons can tolerate better. The field is rapidly expanding, and we now have a whole rogues gallery of promising peptide candidates that have shown success in preclinical studies. Researchers at the Technical University of Munich have developed ring-shaped peptides called macrocyclic peptides that prevent alpha-synuclein from clumping. What's fascinating is that these peptides were originally designed to target proteins linked to diabetes and Alzheimer's. Their effectiveness against alpha-synuclein highlights a potential molecular link between these diseases, which is particularly interesting given that diabetes is a known risk factor for Parkinson's. These peptides work by mimicking a piece of another protein and have been shown to protect lab-grown human dopaminergic neurons from damage. In another approach, scientists used high-throughput screening to test a massive library of peptides. They identified two short peptides, named K84s and K102s, that were highly effective at reducing alpha-synuclein toxicity and aggregation in yeast models. Crucially, one of them, K84s, also worked in human cells, appearing to directly interfere with the formation of the toxic fibrils. Then there's the fascinating area of brain-gut peptides. These are peptides involved in the communication network between our brain and our gastrointestinal system. Peptides with names like glucagon-like peptide 1 or GLP-1 and ghrelin have demonstrated powerful neuroprotective effects in Parkinson's models. They seem to fight the disease on multiple fronts, reducing inflammation, preventing cell death, and combating oxidative stress. In fact, analogs of some of these peptides are already in clinical trials, representing one of the most advanced peptide-based approaches for Parkinson's. Finally, scientists are also borrowing from the body's own protective mechanisms. There's a factor called MANF, or mesencephalic astrocyte-derived neurotrophic factor, that is incredibly potent at protecting and rescuing neurons. Researchers have isolated a small, cell-penetrating peptide from MANF that inherits these protective qualities. Its small size and ability to get inside cells make it a very attractive therapeutic candidate. Despite this incredible promise, the path from the lab to the clinic is filled with significant hurdles. The first and most formidable obstacle is the blood-brain barrier. This is a highly selective membrane that acts as the brain's security system, protecting it from toxins and pathogens in the blood. While essential for our survival, it's a major challenge for drug delivery. Most peptides are too large to cross it on their own. Researchers are exploring clever solutions, like attaching the peptides to molecular Trojan horses that can shuttle them across the barrier, or developing intranasal delivery systems that can bypass it. The second challenge is stability. Our bodies are full of enzymes that are very good at breaking down proteins and peptides. This means a peptide drug injected into the bloodstream might be degraded before it can reach its target in the brain. To combat this, scientists are modifying the peptides, using non-natural amino acids or creating cyclic ring-like structures, like the ones from Munich, to make them more resistant to these enzymes. Third, there's the issue of administration. Because they are broken down in the digestive system, most peptide drugs can't be taken as a simple pill. This means they often require injections, which is less convenient for patients who would need long-term treatment. And finally, the very complexity that makes peptides so specific also makes them more challenging and expensive to manufacture, purify, and ensure quality control for, compared to traditional small molecule drugs. So where does this leave us? While the road to a peptide-based cure for Parkinson's is still long and fraught with challenges, the progress is undeniably exhilarating. The ability to rationally design molecules that can precisely target the root cause of the disease, the misfolding of a single protein, represents a true paradigm shift in how we approach neurodegenerative disorders. The multi-pronged attack, from stabilizing healthy proteins to capping toxic fibrils and leveraging the body's own protective peptides, offers a comprehensive toolkit to fight Parkinson's pathology. As scientists continue to refine these molecules, improving their stability, their ability to cross the blood-brain barrier, and their overall efficacy, the hope for a future with a disease-modifying therapy grows stronger every day. The promise of these tiny peptides holds the potential for a monumental leap forward, not just for Parkinson's, but for other devastating protein misfolding diseases like Alzheimer's and Lewy body dementia. The coming years will be crucial, but for the millions of people worldwide affected by these conditions, the peptide promise offers a powerful and tangible reason for hope.